So thank you very much for joining me today. I've got Andrew with me. He is a senior laboratory technician, which sounds really, really impressive, if I'm being absolutely honest. So tell me a little bit about what it is that you do and impress me.
I run a small laboratory on the actual CRF ward itself. So the ward where the patients who are on clinical trials are staying. The lab is actually physically on that ward itself. So there's a small team of us, as myself, and there's 4 technicians underneath me. And we basically receive every blood sample, every biological sample that is taken from a patient that's required for research purposes. We do whatever is required for it. So the companies in charge will set out these list of instructions beforehand. And they'll say, you know, if you get this sample, do this. If you get this sample, do that. It goes to this lab, it goes wherever. So we'll do all those. We'll do all the paperwork that means that we can track it every step of the way. And we'll either store it in our freezers or we'll ship it onto the lab somewhere else in the world that it goes to. And we do that for every sample, for every patient, for every trial, all the time, basically. So we sit between all the research teams. We work on all the trials, or nearly all the trials. And it's a fairly constant, it never ends. There's never a finite, once we've done this, we're done. There's always something to do.
It's like housework. That never ends.
Exactly.
I can't believe I've just taken your really important role down to housework. But you're always going to be doing something because there's always stuff going on, isn't it?
Yeah. I think the analogy I always use, rather than housework, is more like working in a kitchen, in that you've always got something coming in. Everything's got a different set of instructions to follow. We've got a team of 4 or 5 of us on a day-to-day basis. But on any given day, we could have 20, 30, 40 trials coming in. So there's going to be times where you've got 3 or 4 trials on the go at the same time. So one of them might be really complicated and you need to really focus on that bit. It might be really technical. One of them might be getting samples every hour for the next 8 hours. One of them might be just really easy, or you just need to put this in a package and do a bit of paperwork and then it's done. But you've got 3 or 4 things on the go at the same time. So, you start spinning these plates and you've got to keep them all spinning. So you're constantly prioritizing and keeping everything kind of in range. So it's quite interesting. It's you can't, for all the best wishes, you can plan, you can schedule, but when things happen on the day, you just have to react. And that's, I think, one of the things that I enjoy the most about the job is it's not just at your laptop 9 to 5. It is very much you have to think on your feet, you have to be hands-on, you have to problem solve.
You mentioned about the fact that you're on site.
Mm-hmm.
Is that common? Is that normal, or?
It varies. So obviously the pathology labs, the non-trials ones we were talking about, they would be on every hospital as standard. Doesn't matter how small they are, they have some form of labs. So, because research is so integrated, or should be really so integrated into care, that we do have to kind of say, well, it doesn't matter where it comes from, you bring it to us. It's really to do with the time-sensitive nature of the sample. So obviously, if I've got a sample that needs to be processed within 15 minutes, if you're a patient, by the time you've bled that patient, looked after the patient physically, and then taken that out, doing what you're right, you don't have that time to walk 5 minutes across the hospital. So the closer you are, the better. And it is something that, certainly comparative, early phase CRFs around the country. So sites like Leeds, Liverpool, Newcastle, Marsden, things like that, they will all have similarly located labs to what we have.
How did you get involved in this career? What was it that led you to doing this?
A bit of luck, actually, almost, in a way. So my education, my background is human biology and medicinal chemistry. So I got a job in a hematology lab at Manchester Royal with the aim of going down the route of being a biomedical scientist. By chance, the lab that I worked in at Manchester Royal, the staff there, when you were a supervisor, worked on rotation in the CRF lab at Manchester Royal, so in research there. One year there was a period where we had quite a lot of staff off sick, so I ended up working, rather than 1 week on, 1 week off, I ended up basically doing 2 months kind of solid in the research lab. And I was I quite like this. This is really good. It's very different to the kind of much more automated labs that you would work with in pathology. It's much more hands-on. I said much more reactive. You were quite independent. You were still working as part of a big team, but you kind of had the autonomy to do what you needed to do. And a job came up for a senior in a research lab, a 10-minute walk from my house at the Christie. And I was like, well, I've just literally come out of 2 months doing this non-stop. I reckon I'd have a good chance at this. And that was 5.5 years ago.
You struck on something a little bit there, was that you didn't really know that this place, it really existed.
Yeah.
I feel like people say the lab, and it's surrounded with mystery in general, isn't it?
Oh, yeah.
Or we'll send that to
The lab.
The lab.
Yes.
It's just one of those things, isn't it? You go, nobody really knows where it is, what it is, and what happens there.
You're right. So when people say the lab, people kind of have this image of, two guys in white coats, a microscope in a dark basement room somewhere like that. The labs at Manchester Royal, for example, the pathology department there, that division is 4 floors. The staff-wise, it hires over 750 staff members, nearly 1,000 staff members, and receives samples from like all over the world. Like the lab that I worked in 1 week would go through 35,000 blood samples. The lab that I'm in now is obviously a much smaller affair, but we routinely do between 500 and 750 blood samples a week. People always have this idea of just like it being a little, dark corner of the hospital somewhere in a basement and no one really knows what goes on. But actually, like, labs are huge. And as we've mentioned earlier, kind of like the research wouldn't happen without this lab. Like the samples would get taken. Yes, some of them would get sent off by nurses, but like anything that needs processed, anything that needs stored, anything that needs to be shipped off to the labs, any early phase trials just wouldn't happen without these labs in every hospital. So I think we're very, like, not overlooked in like a self-deprecating way, but overlooked in the fact that a lot of people don't know we exist at all.
So I've been asking everyone I'm speaking to about some of the myths around clinical trials, clinical research. Now, I'm guessing you're coming from a slightly different angle on this, but what would be the myth that you would like to bust about clinical trials and clinical research?
It's difficult. There's a few ones that I always get. So when I speak to my friends about this, so they always introduce me, he's trying to cure cancer. And I was like, that's a very big introduction. And I always end up in these conversations with people who aren't scientists asking kind of like, what is it you're trying to do? And it's not really a specific myth as such, but it's always something that I try and explain as like, there isn't going to be a cure for cancer. It's not how it works. It's like, that's not what we're trying to do. I think the most important part of our job, can we get to the cancer earlier? Can we predict it? Can we predict what it's going to do beforehand? Can we match people with the right trials before we even create the trials? Some of the biggest single trials that we've handled samples for are trials like that, where we'll take samples for patients regularly, and then they'll be doing tests for very specific biomarkers, very specific proteins or things in the blood, and saying, actually, this gets higher at this point, and then suddenly they progress. Can we use that as a test? And that's, I think, the bigger thing. And certainly, like the way I see it, is it's like, yes, great, new drugs, more effective drugs are super important. But the earlier you can get to them, the patients that is, and the diagnosis, and the better you can match them before you even give them a drug, that's where like the real kind of gains are to be made.
Andrew, thank you so much for talking to me today.
That's all right.
Really enjoyed it, learned absolutely loads, and I'm going to let you get back
No worries.
to the organised chaos that is the lab.
Oh, it is, and it is organised chaos at the minute, so